Assalamualaikum and greeting everyone. Welcome to my final year project presentation. I'm Nur Raihana binti Rosli, a final year student in Bachelor of Science Honours Biology. The title of my FYP is the Morphological Characterizations of Entoloma species from Jengka. I'm supervised by Dr. Nur Azliza binti Ismail. This presentation consists of 5 chapters which are Introduction, Literature Review, Methodology, Result and Discussions, and also a conclusion and recommendations. Let's begin with Chapter 1, Introduction. In Malaysia, the study of Entoloma has been relatively limited. Many of the new findings are actually from previously unreported species, as well as newly discovered species which have not been published yet. Entoloma species identified from previous research are identified only on their macromorphological and absence of microscopic observations. The morphological characteristics differ in different subgenus, never been discussed in detail. This is probably due to the undiscovered species and lack of expertise in Malaysia. The significance of this study are contributed to updated information on Entoloma species in Pahang, especially in Jengka, provided additional information on morphological characteristics of Entoloma and the documentation of undiscovered Entoloma species in Pahang. There are 3 objectives in this research, which are to collect Entoloma species from University Technology Mara Jengka campus and Sri Garden Forest, to determine the morphological characteristics of collected Entoloma species using light microscope and electron scanning microscope, and also to identify collected Entoloma species based on their morphological characteristics. Literature review cover on several aspects. There are several characteristics that differ Entoloma species such as basidospores, basidia, hyphae, pilus, and lamella. Entoloma have various subgenus that varies from their characteristics. Subgenus Cloudopus has small basidiums and frequently ignored during data gathering. Leptonia have different hues which usually attractive in black, blue, green, yellow, and violet. Nolania's cap usually in conical, convex, or ambonate and have the angular spores. Cyanula have adnate lamella and lamellula, and Cyanula is the most species-rich subgenus within Entoloma. Pozzarella has the range color from dark grey to brown in their lamella and absence of clamp connection, while Alboleptonia have white to ash grey basidial carp, opaque stripe, and dull pink basidospores. Next, we jump into Chapter 3, Methodology. The site study are in University Technology Mara Jengka campus and Sri Garden Forest. The sampling process are began with specimen collection that collected during raining season in 2021 and 2022. Sample collected were labeled with collection number, date, and place. The sample were observed in site with taking picture and measurement of all specimen collected. The pilus, lamella, stipe, and other features are jotted down. The sample collection then taken to laboratory for microscopic observations using light microscope and electron scanning microscope. The distribution of Entoloma species that are recorded are 1 species in Sri Garden Forest, while the other 6 from Uitm Jengka, specifically founded in Hilltop, College Mat Kilau, Al-Ghazali Building, Merempuh Halangan, and Taman Herba. After thorough observations and research, these 7 species are divided into 4 subgenus, which are 2 from Leptonia, 3 from Nolania, 1 from Cyanula, and 1 from Alboleptonia. The first one is suspected Entoloma hymenidernum. The sporocarp was blue in color with a dark blue in center. The pilus was convex with uniformly oppressed velvety surface. The pilus surface was smooth and slightly wrinkled. Noticeable lamella and lamellula with subdistant spacing of lamella, adnate and attached to stipe. The stipe vivid blue lines alternating with pale blue whitey stripe. The stipe was in clavate form, smooth texture, and twisted stipe. No ring and distinctive odor were observed. The basidospores were angular, heterodiametrical, 5 to 6 angles. Clavate basidia were present, hyphae and clamp connection also present. This species suspected Entoloma hymenidernum as it share the same characteristic, which are fibrillous pilus and stipe, convex shape of pilus, velvety surface, subdistant lamella, twisted stipe, heterodiametrical basidospores, clavate basidia, and presence of clamp connection. Entoloma hymenidernum could be mistaken as Entoloma eugeni and Entoloma subcarnium. This Entoloma could not be identified due to some factors, but in Leptonia subgenus, the sporocarp was appeared peach pink in color, with brown pattern. The texture of sporocarp was scaly, distant spacing of lamella, adnate attached with stipe. The stipe in cylindrical form and has fibrillous surface texture. No ring and distinct odor was detected. The basidospores were angular, heterodiametrical, 5 to 7 angles, clavate basida, septate hyphae, and absence of clamp connection. Species 1 suspected Leptonia subgenus for its heterodiametrical basidospores, absence of clamp connection, similar with research by Karstedt et al. In 2022 and saprophyte. This Entoloma was suspected Entoloma staurospora. The sporocarp was in shining dark brown, smooth and silky surface, noticeable lamella and lamellula with distant spacing of lamella, adnate lamella attached, vivid brown lines alternating with pale grey whitey stripe on stipe, clavate stipe, twisted and smooth surface type, no ring attached. Basidospores were angular, prismatic, 4 to 5 angle, clavate basida, cylindrical hyphae, and absence of clamp connection. This Entoloma suspected Entoloma staurospora as it has dark brown sporocarp with striate cap, unique prismatic shape of basidospores, adnate to adnax lamella, and lack of clamp connection. This Entoloma suspected Entoloma vernum as the sporocarp in conical shape, sheen gleam surface with bronze color, lamellula and lamella with subdistant spacing, pale brown with white lines type cylindrical, twisted type, and fibrillous surface. No ring and distinct odor were noticeable. The basidospores were angular, mostly in cuboidal shape. Have cylindrical basida and hyphae, also absence of clamp connections. This species has the biggest spore among 7 Entoloma found. This species assumed Entoloma vernum for its conical pillars, fibrillous type, and shiny brown to bronze color of pillars. This species also can be mistaken as Entoloma holoconiatum but differ in pillars, resembles Entoloma violaco-vernum but differ in basidospore size, and resemble Entoloma conferendum but differ in spore shape. This species is suspected Entoloma luteodiscum as the sporocarp was yellow in color, light yellow at the center, subdistant lamella and lamellula, stipe in dark yellow, cylindrical form with twisted stipe and smooth surface. The basidospores were angular, heterodiametrical, cylindrical hyphae and basida, and also absence of clamp connection. Suspected Entoloma luteodiscum as possess yellow in color, convex pillars with smooth surface and identical size of spores. This species can be mistaken as Entoloma murrayi but differ in pillar shape and basidospores shape. This species assumed Entoloma cerealium as its sporocarp was greyish blue color with smooth surface, slightly crowded lamella and lamellula, adnax attached with stipe, club-shaped form and smooth surface texture. The basidospores were polygonal, heterodiametrical, 5 to 7 angles, cylindrical basida, cylindrical hyphae, and absence of clamp connection. This Entoloma suspected Entoloma ceruleum as both have blue sporocarp and pillars, same color of gills, and the shape of spores. The last Entoloma is suspected Entoloma sericillum as this species have features of convex shape of pillars, dry and granular surface, slightly packed gills contain lamella and lamellula, adnax attached with stipe, club shape, and slightly fibrillous stem surface. The basidospore were heterodiametrical to cuboidal shape, 4 to 5 angles, clavate basida, and absence of clamp connection. This species suspected as Entoloma ceruleum as both has convex pillars, incurved margin, and angular sporocarp. As conclusion, 7 Entoloma species have been discovered from tree guarding forests and several places in Uitm Jengka. These 7 species came from different subgenus and inhabit soil or decayed wood. Although these species came from the same family Entolomataceae, they have different characteristics. Being in the same subgenus doesn't mean that their characteristics are same. Several limitations in this project are there are that of local reference, which most of report are from overseas. The sample collected are also small as the range received along specimen collection is not much as expected. Numerous animals, including cats and monkeys, can impede the growth of the Entoloma species at Uitm Jengka. Some species are collected before they reach maturity, which has an impact on the micromorphology observations. For recommendations, molecular tests should be done to identify and address the species to their specific species. Alternatively, dried basidocarp extract can be used for DNA extraction and PCR, or primer design and phylogenetic analysis can be done by observing the genome sequences. That's all from me. Thank you for having me.